This series of videos will cover cell respiration and the steps associated with cell respiration. Now realize that living cells are constantly using energy for activities such as movement, protein synthesis, active transport, or cell division. And most of the energy to power the processes that occur in a living body come from ATP. So a cell must constantly be replacing the ATP that the cell uses. And the cell does this by breaking down organic food molecules and releasing energy that is used to join ADP and phosphate to form ATP. The breakdown of food to release energy occurs by two kinds of processes. The processes include cell respiration and fermentation. So as we look at our chemical reaction for cell respiration, we have glucose, which is C6H12O6, as a fuel molecule that's going to be oxidized to form carbon dioxide and water. So we have our glucose and we have our oxygen. Our products will be carbon dioxide, water, and then ATP energy. Energy that's captured through this formation ends up calculating to be 36 to 38 ATPs per glucose molecule. The energy released during cell respiration is used to regenerate the cell's supply of ATP energy. ATP donates energy to various energy-requiring processes such as metabolic reactions, active transport, and muscle contractions. ATP stands for adenosine triphosphate and is composed of the nitrogen base adenine, the 5-carbon sugar ribose, and 3 phosphate groups. Notice that between the ribose and the phosphate we have a bond with a little bit of energy in that bond. Between the first and second phosphate we have a bond, and it has some energy, but the most energy is held in the bond between the second and the third phosphate. So that when ATP is used as an energy source, this bond breaks between the second and the third phosphate, and it releases an inorganic phosphate plus energy in order to do the cell's work. As a result, we have that inorganic phosphate, and we have a new chemical called ADP, or adenosine diphosphate, that is formed. To make ATP, ADP plus phosphate come back together where the phosphate rebonds to the second phosphate, and then we make the ATP. The mitochondria is the site of cell respiration and is known as the powerhouse of the cell since most ATP production occurs here. The parts of the mitochondria include the matrix, which is the open portion of the mitochondria, and this is where the Krebs cycle occurs. The cristae, which are the folds of the mitochondria and where the electron transport system occurs. The inner membrane, the outer membrane, and the cytosol, which is outside of the mitochondria and where glycolysis occurs. So as we look at the stages of cell respiration, it will be important to note the cytosol, the matrix, and the cristae, as this is where the 3 stages will occur. Now here we have a transition electron microscope photograph of the mitochondria. We can see the cytosol area right in here, and this is where glycolysis occurs. We can see the outer and the inner membranes of the mitochondria. We can see the open matrix where the Krebs cycle occurs, and we can see the folds or the cristae, which is where the electron transport system and oxidative phosphorylation occurs. So the 3 metabolic stages of cell respiration that we will be discussing will be glycolysis, which occurs in the cytosol, which is outside of the mitochondria, the Krebs cycle, which occurs in the open area of the mitochondria, and oxidative phosphorylation associated with chemiosmosis, and this occurs in the cristae or the folds of the mitochondria. And we will take these 3 stages and discuss each stage in detail as far as what chemical reactions are occurring in each stage. The first step in cell respiration is glycolysis. Now, glycolysis occurs in the cytosol outside of the mitochondrial membrane. And glycolysis begins with the starting substance of glucose, and you'll notice that we have the 6 circles, and those circles correspond to carbons. So we have a 6-carbon glucose, and it's going to go through a series of chemical reactions, but it ends up forming 2 3-carbon pyruvic acid molecules. Now, to go from our 6-carbon glucose down to our 2 3-carbon pyruvic acids, we need to discuss some of the activities that occur. First off, we need 2 ATP units to begin the reaction. So because ATP is used up, we have a phosphate broken off of the ATP, and we make ADP, and then our phosphates will attach to the glucose molecule, making a fructose 1,6-diphosphate. Now, in the process of going from the glucose down to the pyruvic acid, we've used up the 2 ATP, and then notice that we end up making 4 ATP. So 4 ATP are made, but because of the deficiency of the 2 ATP here, we end up with a net gain of 2 ATP. Also notice that we will have our coenzymes NAD picking up hydrogens and electrons that have been released during the process of these chemical reactions. Now, these coenzymes, these NADHs, will carry the hydrogen and the electrons to the mitochondria, where they can be used in the 3rd step of cell respiration. And so we end up going from a 6-carbon glucose to 2 3-carbon pyruvic acids, using 2 ATPs, making 4 ATPs, so we get a net gain of 2. We have our phosphates being broken off. We have hydrogens and electrons being released. They're being picked up by NADH and taken to the mitochondria. Next, we will take our pyruvic acids, and what happens will be determined on whether or not oxygen is present or not. If oxygen is present, then we are going to move to the mitochondria, to the Krebs cycle. If oxygen is absent, then we're going to move to making lactic acid. Now, making lactic acid is less efficient. So, fermentation here, making the lactic acid, has a net gain of the 2 ATP shown right here. And so it's only about 2.1% efficient. So what we really want to happen is we want to move towards the Krebs cycle. Now, between glycolysis and the Krebs cycle, we will have a transition reaction that occurs. And so we'll discuss this transition reaction on the next slide. So between glycolysis and the Krebs cycle is a transition reaction in which pyruvic acid is converted to acetyl-CoA and 2 CO2 molecules are removed. Remember that glycolysis occurs in the cytosol outside of the mitochondria, and the Krebs cycle occurs in the open area or the matrix of the mitochondria. So the 2 3-carbon pyruvic acids will need to move from the cytosol area into the matrix section of the mitochondria. And as they move, they will join with a coenzyme, and they will lose a CO2 molecule each. And so they lose that carbon, and that carbon joins with the oxygen that one breathes in. And so now we see why an individual must breathe in oxygen. It's so that oxygen combines with the carbon to make the CO2. The coenzyme will then help the acetyl molecule to enter the Krebs cycle. Now, acetyl is a prefix used to describe a 2-carbon molecule. So we have gone from 2 3-carbon pyruvic acids with the help of a coenzyme A. We have a release of 2 carbons, one from each pyruvic acid, that joins with the oxygen that we breathe in, resulting in 2 2-carbon compounds, and these being acetyl. And it's this acetyl that is going to enter into the Krebs cycle. The CoA will release the acetyl, and then being an enzyme, it will go back and it will help the next component to enter the Krebs cycle. Because it is an enzyme, it's constantly helping the reaction begin, but is not part of the reaction in itself. Also be aware that in the process of this reaction, we do have our coenzyme NAD picking up hydrogens and electrons, and it becomes NADH, and it's going to take this material right here, these hydrogens and electrons, to our third step of cell respiration.